Hi everyone, thank you for joining the fifth webinar in our series, Use of New Approach Methodologies for the Risk Assessment of Pesticides. This series is being co-organized by PETA India, the Biological Agri Solutions Association of India, and the Society for Alternatives to Animal Experiments India. I'm Dr. Ankita Pandey, and I'm PETA India's Science Policy Advisor, and I'll be moderating today's webinar. Today, we will hear first from Dr. Bharath BR, who will be giving an overview of the computational tools to predict pesticide toxicity. Before I introduce the first speaker, I would like to point out that information about the webinar series can be found at the link on this slide. There will be time for questions for the speakers at the end of the presentations. Everyone is on mute, but you can type questions or comments in the question section within your GoToWebinar toolbar at any time during the presentation. This toolbar should be on the right-hand side of your screen. Please do mention to which speaker the question is being addressed to. Our first speaker, Dr. Bharath, completed his MSc and PhD in bioinformatics from Kuvempu University, Shivamogga, Karnataka. In his 14 years of experience in computational chemistry, he has closely worked with scientists from different backgrounds like synthetic chemistry, biotechnology, system biology, and computer science in academia, pharma industry, and CROs. He is a recipient of Young Scientist Award from the Vellore Institute of Technology, Vellore, for his PhD work, and has 27 research publications in high-reputed international journals and an Indian patent too. Dr. Bharath currently works as Senior Research Officer at J Research Foundation, specializing in silico toxicology. It is now my pleasure to turn the floor over to Dr. Bharath. Dr. Bharath, you're on mute.
Sorry, am I audible now? Yeah. Hi, Ankita. Thank you very much for giving me an opportunity. And I would like to thank BASA as well as PETA India for giving me an opportunity to share my thoughts on computational tools to predict the pesticide toxicity. And I would like to introduce our organization, J Research Foundation. And we are actually a preclinical CRO located in Vapi, Gujarat. And we have a GLP facility where we offer a wide range of GLP-based toxicological preclinical studies. And today I'm going to talk about computational tools to predict the pesticide toxicity. And hope we are good to start now. Yeah, thank you. And I would like to give a bit background on today's discussion. As we all know, EPA US or US EPA has the Toxic Substance Control Act, and it was charged to evaluate the toxicity of over 85,000 different chemicals that are produced, imported, or managed in US. So the reason behind this is every chemical which is going to be produced or imported are supposed to be evaluated for its toxicological effects. When we have to evaluate these 85,000 plus molecules, and certainly it is going to be very expensive, time consuming, as well as ethically challenged. And apart from that, it is not effective in keeping up the same pace of constantly growing chemical inventory, because as we know, good number of chemicals are coming or emerging day by day. So evaluating all of them for different or wide range of toxicological parameters is going to be really challenging with respect to time, cost, as well as ethical constraints. So US EPA has also got a sense of it, and it has started promoting the cheminformatics based open access computational toxicological tools, and it has also shown interest in developing the tools which can supplement and certainly replace the in vivo bioassays. So the total objective of this effort was to minimize the usage of animals. So here you can see the dashboard of CompTox, and you can see the number here, and it is holding around 12 lakh different chemicals in it. So I have captured this screenshot on 29th June. So if you check today, probably there may be 3 to 4 new entries. So this is how constantly the chemical inventory is growing. So keeping that in mind, actually the US EPA has taken initiative in promoting the computational models for the prediction of wide range of toxicological endpoints. And this particular Comptox chemical dashboard actually provides multiple models for the screening of chemicals, and it helps us in prioritization, hazard identification, as well as determination of a mechanism of action, and it certainly helps us in risk assessment. And when it comes to the regulatory needs from consumer perspective, because we all are consumers, we always look forward to have a safe products or safe pesticides which are not that toxic to the environment as well as for the human or mankind. So when it comes to the impacts of these pesticides, they are used in our countryside, urban areas, homes, as well as gardens, and they have their negative impacts on our health. And when it comes to pregnant mothers and children, this is very vulnerable group, and they are very susceptible. And it also drains our economies because of the usage of the pesticide, people go sick, and it causes a significant effect on productivity of individuals, farmers, and all. So it leads to the negative impact on our economy of a country also. At the same time, it affects on a biodiversity, because when we use a pesticide, it is going to get exposed to not only the target of our interest, like whether it is a pest, herb, or anything. It is going to be released into water bodies, and it will be released into the environment. So the living organisms in the environment may be negatively impacted by these herbicides or pesticides. So ultimately, it is going to cause a negative impact even on the quality of the soil as well as air. So this is how there is a need for controlling the toxicity of these pesticides. So from consumer perspective, at the same time, when it comes to the resistance perspective, resistance also want to develop a safe products at the same time, and they are also being controlled by multiple regulators. So we have a list of pesticide registration or regulators, and different acts to regulate the untoward effects of these pesticides. And this is applicable for use and sale, and these are regulated by multiple bodies as well as acts. So we have popular acts like FIFRA, we have a FDA, we have a Food Quality Protection Act, we have a Pesticide Registration Improvement Act, and we also have a 40 CFR. And these are global, and at the same time, there are some specific laws and regulations regarding pesticide registration, and they are very much applicable within a states, also within a particular state. So when it comes to use and sales, so this is another challenge for a resistant. If they want to go for a multiple states in a US, then they will have to satisfy, or they are supposed to be abided for these regulatory requirements. And when it comes to India, we also have a CIBRC. It is actually regulating a usage of pesticides in India, and they also have a comprehensive registration of pesticides starting from use to sale, and even it is focusing on import of pesticides. And when it comes to the pesticides and different regulatory challenges, as I have mentioned, there are multiple negative impacts of these pesticides, and the resistants are also expected to submit toxicological reports, or to prove their product is safe for health effects as per the test guideline studies. And I'm just listing a few toxicological endpoints, like a skin sensitization, eye irritation, carcinogenicity, genotoxicity, developmental and reproductive toxicity, endocrine toxicity, neurotoxicity, immunotoxicity. Likewise, we also have an ecotoxicity, environmental fate assessment. Like this, we have a range of toxicological endpoints, and these are going to be varied from product to product and utility too. And when it comes to the predictive chemistry, as we know, to evaluate the toxicity of a given test item for uh, these all endpoints, you just imagine the cost as well as the time required and animal involved in this, and it is too much for one particular compound. That's why even nowadays regulators have started accepting the predictive results from the in silico or computational tools, and these tools can be used when the ability to generate a new data is limited by cost, time, or regulatory framework. And in silico predictions can always support the risk assessment, and they can add the weightage of evidence to the known endpoint, because we have a couple of incidences where the result will be ambiguous. Just to take an example, when it comes to skin sensitization, there are multiple assays, we have an in-chemico assay like a DPRA, and we have in vitro assays keratinosin assay as well as HCLAT assay. If the results are ambiguous, then as per OECD 497, the in silico data is acceptable. So we can add a support of evidence or weight of evidence to the results obtained in vitro or in chemical. So this is how we can also add a weight of evidence to the particular toxicological endpoint, and the uncertainty in a toxicological prediction can be reduced. And we can also use it as a screening platform, not only keeping the regulatory requirement into consideration. If you have 8 to 10 molecules, and instead of taking all the 8 to 10 molecules into the pipeline, we can screen them before we take them into the pipeline. So these in silico methods should be well documented to maintain the transparency, so that we can take the regulators into confidence. So by the time we go for the prediction, applicability domain should be very well understood based on the data on which the model is actually built. And most of the time we will be using the used model or developed models, and before using a model we are supposed to pay attention on understanding the applicability domain of the model what we are using and for the particular utility. So when it comes to the proper flow work, we will have to collect the known data for risk assessment, and this known data should be very relevant for the toxicological endpoint or for the product which we are going to expose. And what is the exposure estimation, and what are the kind of the exposure expected to the given pesticide or insecticide, and what kind of the hazards can be predicted, and what kind of the endpoints are really relevant for this risk assessment. We are supposed to characterize them, and after that we will have to look for exposure refinement. So all these things, if all these things are well placed, we will be in a position to come out with the proper risk assessment conclusions. So as there is a popular quote by Robert Kiyosaki, the best way to predict the future is to study the past or prognosticate. So here we have a techniques like QSR, and sometimes it is a quantitative structure activity relationship, and in some instances we will have to be satisfied with the qualitative structure activity relationship too. And here we will be using the known molecular information to predict the new outcome or for a new molecule. So this is how we'll be using the known data. So there are different data bases which are providing enough information regarding the molecule, regarding the toxicological endpoints, regarding the ADME endpoints, or multiple data points are available in databases, and it's all up to us to learn from these past experiences deposited by multiple researchers, and how better we prepare for the future for the prediction of a molecule. And this is how I'm just trying to give a basics on this QSR, and this is again a computational approach. Here we start with acquiring the chemical data. So you can first go for acquiring the data or curating the data, relevant data very particularly, and this data would be either chemical centric or activity centric. So the response data has to be clearly mentioned or clearly decided before we start the curation, and chemical information should be properly covered, and the redundancy as well as unwanted data is supposed to be removed by cleaning the data. And this is again a mathematical model, and here most of the time it's going to be a regression model or a categorical model. So the suitable method has to be selected, statistical method has to be selected. We have multiple algorithms for developing this mathematical model. So suitable model has to be selected depending on a type of a data set we have for a prediction. And once we come out with a model has to be statistically validated, and this validation is supposed to be both internal as well as external validation, and interpretation of this information should be unbiased, so that we will come out with a better prediction or a design for a prediction. So here, when it comes to QSR, there are multiple dimensions starting from 0D to 7D, because to what depth we are looking for the prediction, and depending on that, we will be in a need to go for a selection of this dimension of QSAR. So just to give a quick introduction to this, we have 0D, where only the chemical formula of a molecule will be taken into consideration, and it is not that reliable. And we also have a 1D QSAR, where substructural fragments or R group information will be mostly taken into consideration. In a 2D QSAR, most of the tools, whatever we have now, are working on the basis of 2D QSAR. These all are topology derived, and how the two-dimensional or structure information is basic over here, and on the basis of that the prediction will be made. And when it comes to 3D QSR, the binding posture of the molecule to the given target and its occupancy in a given 3D surface, as well as its spatial geometry, will be taken into consideration. And when it comes to 4D QSR, the conformation active pocket influenced by multiple energy factors, orientation, protonation states at different pH. Like this, we will go on adding one and the other dimensions. And when it comes to 5D, here in a 3D and 4D, the protein will be rigid, only the ligand will be flexible. But when it comes to 5D, we will have an active pocket of a protein which is flexible, and this is adding another parameter called as induced fit parameter. And when it comes to 6D, not only the flexibility, when it is in a solvated state, how does it works? And when it comes to 7D, real target based receptor model data will be taken into consideration. So each and every dimension of QSR are added with multiple parameters. Just to have a better understanding, I have a simple analogy here. So 0D or 1D are similar to the regular screen in a theater, and whereas these 3D to 7D are multiple features which are going to be added to this theater, where we will have a bubble effect, snowing effect, and we will have a movable chase, so that we will have a 3D as well as 4D effects in a visualization. So similarly here also we will go on adding multiple parameters to increase the reliability of the prediction and to meet the realistic requirements. And here the major thing we have to understand in QSR is descriptors, because these are the determinants of our prediction, and at each level of QSR we have multiple descriptors. When it comes to 0D descriptor, we have a constitutional indices, molecular property, atom, as well as how many bonds we have. That is going to be a parameter. But when it comes to 1D descriptor, we will have a fragment counts, how many fragments, how many R groups, and what is its fingerprint. All these things will be taken into consideration. Whereas in 2D descriptors, we will have a topological considerations, bond angle, bond length, everything will be taken into consideration, and what kind of a physicochemical parameter the molecule is having will also be taken into consideration. And as well as to be particular with the 3D descriptor, electronic parameters, spatial parameters, molecular shape analysis, molecular field analysis, receptor surface interaction, and many more other parameters will be taken into consideration when it comes to 3D descriptors. So in each level, descriptors are going to be increased and their complexity level will be increased, so that we will come out with the meaningful predictions. And just to give a typical process of QSR modeling, as I have mentioned, we will have to go for a sourcing of our data, and we have two phases here. First phase is a development stage, and the second one is apply and validation stage. So first we will have to source the right data, and we have multiple databases for this chemical information, and we will have to curate them, and after that we will have to partition them into training as well as test set, because it is always necessary to have a good quality training as well as test sets to have a meaningful model, and we will have to develop the model. Usually we partition the data with a ratio of 7:3. 70% will be in a training set and 30% will be in a test set. And once we develop a model, we are supposed to validate them internally as well as externally. When it comes to internal, we take this molecules from the same data set and we will validate for their reliability of prediction. External means we take new molecules which are not there in a data set and we validate them for the prediction. And here the activity will be known for the validation. And we will have to run the new data, more and more data, to gain more and more confidence. So once we get confidence on the model developed, we can go for a usage of this particular developed model for the prediction of new molecules. So after QSR, we also have another approach called as read across approach. Here, if there is no proper endpoint specific entry in our data set, then how better we can fill this data gap. So it's an alternative approach to fill the data gap for the substance, and we usually call our molecule to be predicted as a target for a particular endpoint by using the data from another structurally known or mechanistically known molecule. So this is how the read across works. And here you can see the reliable data point, and this is going to be a source for the prediction of a missing point. So we can predict the defined endpoint for this particular target by taking a reference of source molecule, which is either structurally or mechanistically similar. When we are going to take data, or when we fill this data gap from two different source points, and by then we can call it as an interpolation. At the same time, two different source points will be used for the extrapolation of data for two different targets. Then we call this as a endpoint extrapolation. And to have a better understanding on it, here we have a simple graph, and we have a carbon chain length. It is defining as a structure of a given molecule, and we have a toxicity value in y-axis. And this is where our molecules or analogs in a model are fitting. And here you can see, if we actually take the data from two different source points to predict the activity of a given target, then we call it as an interpolation. At the same here there will be no relation between the molecules in a read across. So this is where we have two different approaches, like analog approach as well as a category approach. In analog approach, the extrapolation usually works, and in a read across actually category works. And we can also have some outliers which are not fitting in the curve, so we try to avoid them during the development of a model. So if we have a new molecule, if you want to identify or predict its activity or toxicity, we can take the data from the known source data point and we can predict it. So this is again an extrapolation approach. So there are multiple approaches again in read across, like one-to-one read across, where one source point will be taken into consideration, whereas in many-to-one read across, we will take data from multiple sources. It is again interpolation. So as I was mentioning, multiple source substances will be used, and there is no similar trend or pattern in their properties, then we coin them as the analogs. And if there is a similar trend or pattern in their properties, we call them as a group or a category. So this categorization and subcategorization will certainly help us in coming out with a meaningful prediction. So here we can see when it comes to the prediction of this particular target, we are going to call it as a C8 to C14 because we are using the data from C8, C10, C12, C14 as well as C12. So the coverage is very high. That's why we are going to predict a toxicological parameter of this molecule with reference to multiple source points. So this is the classical example of again many to one read across. And when it comes to read across process or a framework, again we will have to define the decision context and where exactly the data gap is there. We will have to analyze it, and we will have to profile the target to which particular class of chemicals my target molecule is going to be. And we will have to go for a target alert search, so where we will be identifying the source. And if there are proper alerts for our targets, then we will have to continue for the source identification. Otherwise, we will have to exit the read across at this point. And if you get the good sources, then we will have to evaluate the obtained source, whether the obtained source is really of good quality. If the quality of a source is good, then we will have to continue with the source data evaluation. Otherwise, we will have to exit the read across here. And this is how even the auto workflows work. And if the source data evaluation is really acceptable, then we conclude that read across is really of good quality. If not, we will have to go for identification of a new source, and we will have to keep it in multiple iterations. And there are multiple tools for the QSR as well as read across based predictions. We have I have listed a couple of tools here. We have OECD QSR toolbox, Vega Hub, Opera, Direct Nexus, Sara Nexus, LeadScope, Multicase, Case Ultra, Toxtree, Danish QSR database. I was able to list a few here, and there are many more. And at the end of my presentation, I have listed all the tools, and whoever is interested can go through. And when we go for a selection of a model for our QSR based prediction, and we are supposed to know whether it is regulatory acceptable. And you can see here we have certain guidelines already in place. Just to mention a few: ICH M7 guideline on mutagenic impurities, and we also have a IATA guideline as well as for OECD 497, as I have mentioned. So to satisfy this guideline requirement, we can use multiple tools. And here I have just mentioned few tools. I want to take the name of Direct Nexus, and it requires a license or a subscription. We also have a OECD QSR toolbox. It is freely available. LeadScope again requires a subscription. Vega Hub, it has a multiple endpoints. You can see multiple range of endpoints starting from mutagenicity to DART. I have considered a few endpoints because I cannot consider all the endpoints in my single plot. So I have considered a few from mutagenicity to DART, and it's an illustration like these tools have wide range of endpoints. And we also have a Toxtree which has endpoints for mutagenicity, skin or eye irritation, skin sensitization, carcinogenicity, and all, but it doesn't have endpoints for DART. So like this, we will have to look for the availability of an endpoint in a tool, and we will have to choose the right model. So the quality of a prediction always lies on the selection of a model. So the selection of a model will be always reflected in a applicability domain. So it is always necessary to go for assessing the reliability of our prediction. So when it comes to reliability assessment, the major parameter is applicability domain, and it depends on the training set used to develop the model. Say for example, if I want to predict the thyroid peroxidase inhibitor activity, then I will have to have a thyroid peroxidase inhibitor in my data set, and so that it is going to define the proper applicability domain. So whatever the prediction I'm intending to do, whether my data set is designed for the same or not, we will have to check that applicability domain. And is there any uncertainty in the prediction, we will have to assess. And this is going to this expresses the limitation in knowledge or lack of data in a given data set or a model. and it can be reduced or eliminated by making some changes to the data sets. And when it is a third-party tool, we will not have a provision to change the data set, but when it comes to custom-made QSR models, definitely we can achieve it. And variability is another parameter which is very much essential to assess the reliability of the prediction, and it refers to inherent heterogeneity in the data, and it cannot be reduced, but it can be characterized whether there is a variability or not, and accordingly we can give a weightage for our prediction. Validation is another part where it allows us to evaluate the predictivity and reliability of the model, and as I have mentioned, it can be internal or external. Usually we evaluate with the known molecules to have a validation of a given tool or a model which we are intending to use. And when it comes to reporting, the models, whatever we are using, are reported in a QMRF format, which is harmonized template, and it is structured according to the OECD validation principles. And in case if you are coming out with a new model developed, and we will also have to submit the QMRF according to the OECD validation principles. And the most important thing is to be transparent about the prediction, what it can tell and what it cannot tell. And these particular computational predictions also have some limitations, but these are limitations it can be addressed effectively. And dependency on the data availability is a major limitation, because as I have mentioned, we are going to establish the knowledge base. On the basis of that, we will be doing the prediction. If there is no proper knowledge base, it will be tough for the prediction. Analyzing or prediction of toxicity for multi-constituents or formulations is tough, but currently we have some approaches where we will consider them as the monoconstituents and we do the prediction, and their percentage in a given formulation will be taken into consideration. Another limitation is prediction for the UVCBs, particularly hydrocarbons. So this is because these are going to be molecules which are very limited in our data sets, and UVCB data set is difficult to do the prediction, because most of the molecules will be looking similar structurally as well as with respect to the functional group. So that is another challenge we have. And to summarize my discussion in last 20 minutes, in silico models can be used to predict toxicity. The right approach or a model need to be used for the reliable predictions. And during the prediction, make sure that you know the identity of the target chemical, and choose the model, appropriate model for both target chemical type as well as endpoint of interest. Check the target chemical, is it within the applicability domain or not? If not, try to find the suitable model. With all these considerations, we can always use computational models to reduce the time, cost, as well as animals. So I wish you all a happy prediction. Thank you. And these are the services we are offering, in silico services, especially in our facility. We are offering different predictive toxicity, starting from physicochemical parameters till ADME we are offering, and we also have a ligand-based drug discovery services, structure-based drug discovery services, and we also have a platform to identify the mechanism of action of a given pesticide. It may be insecticide, fungicide, or herbicide. We also have a services offered for a protein modeling. And apart from in silico, we also have in vitro capabilities starting from skin sensitive. Excuse me. Corrosion till ADME. Thank you.
Thank you, Dr. Bharath, for such an informative presentation. It was great to know how many of these QSR tools are, freely available, because that is one of the concerns that the industry has, that using these kind of NAMs may sometimes just add on to their cost. So thank you for sharing that information with us. So our next speaker today is Dr. Varun Ahuja, who would be presenting on the use of such computational approaches for genotoxicity assessment of pesticides. So a little bit about Dr. Ahuja. He is a veterinarian and an American board certified as well as European registered toxicologist. He accomplished his graduation in veterinary sciences and master's in veterinary pharmacology from Jimmy Pant University, Panagar, Uttarakhand. He completed his PhD from the Department of Veterinary Medicine, Freie University, Berlin, Germany, and later on postdoctoral work from Charité Medical University, Berlin. Dr. Ahuja started his industrial career working as a study director at BSL Bioservice Scientific Laboratories in Munich, Germany. Later on, he was associated for more than 10 years working in the field of drug discovery and development at Lupin Pharma, Pune. With a vast experience in drug discovery and development, regulatory toxicology, and risk assessment, presently he is working at Syngene International Limited, Bangalore. From a scientific career spanning more than 20 years, Dr. Varun has about 100 publications and presentations. He has presented scientific papers, delivered talks in various meetings in India, Europe, and USA. Dr. Varun is also involved with organizing various lectures, courses for development of students, as well as professions. It is now my pleasure to turn the floor over to Dr. Ahuja.
Thank you, Dr. Ankita and PETA India and the other organizing committee who gave me an opportunity to speak on this forum. So it's really an honor for me. Also, thanks to Dr. Bharath who gave such an exhaustive presentation on QSAR. So he has really made my job very easy. So I can really focus on the practical approaches, how to use various QSARs, because you people already know about the background of QSARs and where they can be applied. So starting with my presentation, so my lecture outline will be a little bit of review of literature, although my lecture will be focusing more on the demo of various in silico tools and some of the work what we are doing in this domain, and how we can collaborate with other scientists who are also working in this domain. So also, although Dr. Bharath has very well explained about the QSAR model, so most comprehensive guideline which is currently available for applying QSAR analysis is provided in the REACH guideline, which was published in 2008, and it says that, just a minute. So it says that whatever QSAR model you are applying should be scientifically valid model. It should be model applicable to the query chemical. So for example, if you are using pesticides, which is actually the topic which we are discussing today, so the software should have the database for the pesticides. So some of the softwares are more inclined towards the pharmaceutical database. Some of the softwares are more inclined towards the chemical or pesticide database. So you should see that the database should be relevant, and also the model should be relevant to the regulatory purpose also. So then only you can apply for you can use that particular QSR model. So Dr. Bharath has already presented a list of various QSR models which are available. Some are licensed software, some are freely available. So you have to see which software or QSR model is more relevant for your purpose. Although I know most of you know about genotoxicity, so I just want to give a very simple definition for this, so people who are new into this domain. So you know, any hazardous substance, it can be a chemical, physical, or a biological agent. When interacts with the genetic material, it can cause genomic instability or mutations, and this leads to genotoxicity, which can be of various kinds, like mutagenicity, DNA damage, or chromosomal damage. And this genotoxicity ultimately can lead to cancer. So today, my presentation will be focused more on the genotoxicity assessment using in silico tools, because this is the area where most of the in silico tools have been validated. Although I will talk a little bit about our other toxicity endpoints also. And you also know very well that for the genotoxicity testings, there are various assays available for mutation testing or DNA damage or chromosomal damage. and people know the names of all these tests very well, like AIMS, MLA, Comet assay, chromosomal aberration, or micronucleus. So I'm not going to talk about these assays. I'm going to talk about how we can use QSR models as a replacement to some of these assays. So I'm sure most of you who are working in this risk assessment domain or QSR domain know this, but for some of the people who don't know this, so there is a guideline published by EFSA, which is named as the Guidance on the Establishment of the Residue Definition for Dietary Risk Assessment. And this guideline covers all the crops, livestock, fish, and processed food, which is a part of the diet. So any and this is just a representative figure or diagram from this guideline, which says that any metabolite from pesticide in this case, if suppose this is not having any data, then you can use QSAR prediction or read across for the genotoxicity profiling of this chemical. And this is accepted by the European agencies very well. So over the last few years, hundreds and thousands of reports have been submitted to European regulatory agencies using QSR models, and this is available on the public domain, and you can go through that information also. So this is just a few statements from the same guideline, that the genotoxicity assessment should be assisted by application of QSAR and read across of the metabolites. So you know that there can be numerous metabolites for any pesticide, and doing assays, which can be in vitro or in vivo assays, costs a lot in terms of money and as well as time. So if you can use some quick screening like computational approach, then it will save you time as well as money. So EFSA accepts that, and it also further says that use of computation models for predictions of genotoxicity should not be based on the use of any single model alone, but on a weight of evidence approach, including all available information provided by the models. To maximize the sensitivity and specificity of the prediction, at least two independent QSR models, where possible, should be applied for each genotoxicity endpoint, including both knowledge-based and statistical-based models. So people who are working in the risk assessment domain, they already know that a combination of two models, that is, one is a expert rule based model and another is a statistical based model, is already accepted in various industries. Like in pharmaceutical industry, there is the ICH M7 guideline, which accepts that you can use a combination of two models for the pharmaceutical impurities, and regulatory agencies accept the data for that. Similarly, here it is mentioned that for pesticides also, you can use these a combination of two models for the genotoxicity prediction. And similarly, after some of the slides, I will show you there is a guideline for the extractable and leachables also, PQRI guideline, which also mentions the same thing, that you can use QSR models, which includes a combination of the two models for genotoxicity prediction. So the conclusion is that QSAR models are well validated for genotoxicity prediction right now, and you can confidently use them for genotoxicity prediction for any chemical. I can't go through the whole guideline, but I'm just showing a case study from the same guideline. So you can later go, this guideline is available in the public domain, so you can just search it on Google and you will find it. This guideline is about 100-page guideline and mentions 3 or 4 case studies where they have very well explained how QSR tool can be used for pesticides and their metabolites. So here they have shown a case study where isoproterenone and their 12 metabolites were analyzed using QSAR and read across. So for the QSAR prediction, they have studied two endpoints for genotoxicity potential, that is gene mutation and chromosomal aberrations. And they have used 4 in silico models, which includes Vega, Direct Nexus, and Toxtree. So I will show you these use of these softwares also in a few minutes. And then for the read across, they have used OECD Toolbox. I will not be able to show you the demo for the OECD Toolbox because this is a very heavy software. So this has to be installed in a PC with a very good configuration. Otherwise, the normal laptops, they just hang up if you run OECD Toolbox on a normal laptop. So I will be showing you a demo of various computational tools. First, we can start from one of the tool. So just inform me if my screen is not visible at any time. Yeah, so starting with, we can use start with the talks tree. So Toxtree is a software which is freely available on the internet, so you can download it. And I will just run a test compound, which is endosulfan. For most of the software tools or in silico tools, you need the smiles and or the structure. So if you have the structure, there is always a provision to draw that. Otherwise, if you have the smiles, you can just copy the smiles and you can run it. So the software will, sorry. Okay, I have copied the bracket also. So once you run it, so software will draw the structure and the smiles are there. So then in Toxtree, you have to select what are the endpoints you want. So one is the Kramer rules. You know the Kramer rules. So they classify the compound into class 1, 2, 3, and accordingly the low, intermediate, or high toxicity. So it's a very simple software, but you need some effort. So every time you have to select the endpoint and run it. So like now you got the prediction for Kramer class. So then you have to move to the next endpoint. So suppose now you want to run in vitro mutagenicity. So you have to select that and again run the prediction for that. So now you have got the predictions for that also. So similarly, here is a summary for that. So once you have that, you can select all the endpoints one by one and then keep on running them. So suppose I select the skin sensitization now and then run it. So it gives the thing like this. And now if you want a complete report, you can save it. So I will just save this report on my desktop so that I can show you how the report looks like. So it says file saved. And here is the report. So here is the report. So QSR is a very simple software. The report is not easy to read, I would say, but because it is freely available, so you can use it for your purpose. So here, so like here we ran it for skin sensitization. So it is saying skin sensitization, yes. Then we ran it for mutagenicity. So it is saying no. We also ran it for Kramer class. So it is saying Kramer class high, class 3 toxicity. So this is how the report looks like. It is a very simple report. Only thing is you have to see what is the information you want from here. Then we go to the next software. We can run Vega now. So meanwhile, it uploads. So I can show you some of the documents which I was mentioning. So this is the EFSA report, and this is the complete case study which they have described here. So the parent compound and the metabolites, and then how they have used QSAR tools to analyze that. So I cannot present that because of the lack of the time. Then there is this JRC 2011, a framework for assessing in silico toxicity predictions case study with selected pesticides. So they have also given these case studies where they have taken various pesticides and then analyzed them using various softwares, especially for the mutagenicity. All the softwares like Direct Caesar, Toxbox, Laser, Topcat, Hazard Expert, and Toxtree. So this is really giving some good information if you want to read that. And then there is the ICH M7 guideline, which I was mentioning. So it also says that a computational toxicology assessment should be performed using QSR methodologies that predict the outcome of a bacterial mutagenicity assay. Two QSR predictions methodology that complement each other should be applied. One methodology should be expert rule based, and the second methodology should be statistical based. So you know that in pharmaceuticals, QSAR is very well accepted for the mutagenicity predictions. Then there is a PQRI guideline, safety thresholds and best demonstrated practices for extractable and leachables. This also says that if insufficient mutagenicity data are available, an in silico assessment can be performed as per the ICH M7 guideline. If the chemical is not a potential mutagen because it does not contain a structurally alert functional group for mutagenicity, the leachable is qualified with respect to mutagenicity. And yeah, so now moving on to the software. Yeah, so we use the same compound, endosulfan. This is Vega software. You just enter the smiles and okay, again. And then the next step is you have to select what are the models or the endpoints you are trying to screen. So here is a list of all the models, so you can select whatever you want to screen. I will not select all right now because it takes a long time, so I will select only one just to show you. So here it's all available: mutagenicity, developmental toxicity, carcinogenicity, acute toxicity, LD50, skin sensitization or skin irritation, eye irritation, chromosomal aberration, micronucleus, estrogen receptor effect, androgen receptor effect, thyroid receptor effect, glucocorticoid, thyroperoxidase, endocrine disruptor, NOEL, LOEL. Kramer classification is also there, hepatotoxicity, BCF, acute toxicity, then aquatic chronic toxicity, mode of action, terrestrial acute toxicity, sludge toxicity, zebrafish, biodegradability, persistence in soil and sediment, in water, in air, octanol, water partition coefficient, water solubility, vapor pressure, melting point, hydrolysis, Henry's law constant, etc. So you can select any of these endpoints from here, or you can just filter the models, whatever you need here from like human toxicity, ecotoxicity, etc., and then select that. So I'm just showing you the report for one of the endpoint, which is mutagenicity. So then you go to the next step, which is export, and you can select on the PDF because it is easy to read as compared to the CSV file. And I am just saving it on my desktop. And then you run the prediction. So it takes around few seconds because right now we have selected only one endpoint. So here is the report, what it looks like. So this is a more easy to read kind of report from Vega. So they give you the prediction summary, whether it is mutagenic or non-mutagenic, along with the structure, SMILES which we ran, and reliability. And then it gives a little bit details that what are the similar compounds where the results are already available with predicted and experimental values along with the CAS number. So you can go and search in the Google what are these compounds actually, and then you can understand the basis of the prediction. It also gives the applicability domain. So you get a really good knowledge, understanding about how the software is working. So this is how the Vega works. I have used only one endpoint, so this report is very short. If you select all the endpoints, so then this report goes into 100 pages or so. So we go to the next software, which is Derek and Sarah Nexus from Lhasa, UK. So this is how the software looks like when the window opens. I find this is a little bit easier to use. So if you have the smiles, you can use smiles. If you know the structure, then you can draw the structure here along with all the toolbars available here. If you already have the file from the chemist, structure file, you can also run that directly with the, if you have the, you can save the file on your computer and run it from here. So right now I am having the smiles, so I will just use the smiles here. I don't know if I will just show you how to get the smile. Suppose if you don't have the smile. So if suppose you don't have the smiles for some particular compound, you can just search on Google for, I'm just searching for a drug. So you can go to the PubChem website. There are a lot of websites, but PubChem is the most easily available where you can get the smile. So here, the smile for most of the chemicals or compound drugs are available. So here is the canonical smiles which you have to run. You can also run this INCHI in the direct. I can just show you how for a chemical also. So it's also available on PubChem, so not a problem. So this way you can search for the smiles if you are not very good with drawing the structures, or you can copy the INCHI which I just showed you on the PubChem website, or if you have the mole file from the chemist, so you can use that also. So once we have this here, so the software will draw the structure by itself, and you have the structure with you. You can then run the prediction. So right now I'm running the direct prediction. So we have two softwares, Derek and Sarah. So Derek gives the prediction for about 40 toxicity endpoints, depending on the compound. So here it has given the prediction. And if you want a complete report, then you can have a complete report also. So I will open the report afterwards. So then I can show you how the SARA runs. So SARA gives the prediction only for mutagenicity endpoint. So I'm running SARA now. The structure is still there, so it gives you the predictions. And again, if you want to get a word report, you can get a word report fast. I will open the report afterwards. And the other thing I was telling about the ICH-M7 report. So ICH-M7 report was for pharmaceuticals, but the PQRI guideline for extractable and leachable also mentions the same term because it means that you are using a combination of two softwares. One is expert rule based and the other is statistical based to predict mutagenicity of the particular compound or chemical. So I'm just running the ICH-M7 prediction. So now when Derek and Sarah will run together, it will give the prediction only for mutagenicity. Although Derek gives the prediction for 40 tox endpoint, but when they run together in the ICH-M7 mode, they give prediction for mutagenicity only. So here you say that the combined prediction is there. Derek says that it is inactive, and Sarah also says that it is negative for mutagenicity. So this way you can get a report fast. And if you want to generate a ICHM word report, then you can also get it. So now we have run all the three, Derek, Sarah, and ICH-7. So Derek looks something like this. It gives you the structure, smiles, the toxicity prediction for various endpoints, and then endpoints where there were no alerts found for other toxicity, and then it gives you the complete alert descriptions along with the reference literature. So if validation comments, so if you want to understand the whole report, then you can just go through whole their report. Then all the compounds from where it generated the prediction based on from the database. So it gives all the data. So it is very good for understanding, and this kind of detailed report is not available in the freely available tools. So similarly, if we go for the SARA prediction, so as I said, SARA gives only mutagenicity prediction. So it will say only for mutagenicity what is the prediction, and it also gives the all the alerts which it found it in the database and the hypothesis, etc. And if you see the ICH-M7 report, which is a combination of Derek and Sarah both, so it gives you the results both from Derek as well as Sarah for mutagenicity, which I showed earlier also. So Derek says it is inactive and Sarah also says it is negative. And then it gives you the glossary also, all the terms used in the prediction. So in this way, I find that that these softwares are easy to use and understand also, because the software gives all the information. I think I have given the demo for ToxTree, Vega, ICH-M7, Derek, and Sarah. ComTox chemical dashboard is it's available in the public domain, so you can easily use it. So I think most of the people here also know how to use it. So Dr. Bharath has already shown this. So you can just type the name of the compound which you are looking for. And it gives you all the information which is available there, all the data, physical, chemical properties, environment, toxicity, etc. So you can just go through that one by one and see what is useful for you. So it is a very good compilation of all the databases in one place. Then coming back to the presentation, so I'm over with the demo. So we also do a lot of in silico work, and this work is done by my colleague Dr. Gaurav Ahuja, and he has submitted this to the Eurotox conference, which is to be held in September 2023. So this is quite relevant to today's talk. So I thought maybe I can present a summary of this. So what he did was he did in silico evaluation of several agrochemicals, including inorganic metals, carbamates, chlorinated hydrocarbons, pyrethroids, organophosphates, insecticides, fungicides, herbicides, fumigants, etc. And he used three softwares, which included QSR toolbox, tox tree, and I already gave you the demo for the tox tree, and test by US EPA. And he chose about 60 agrochemicals, and he did the in silico analysis for various tox endpoints or genotox endpoints, including aims, in vitro mammalian cell gene mutation, in vitro micronucleus, in vitro CA, in vivo micronucleus, in vivo CA, rodent CAR-C, and skin sensitization. Just a summary of the results. So we found very good predictions with some of the tools, and for some of the toxicity endpoints like QSR toolbox, tox tree, and test had accuracy of about QSR toolbox had accuracy of about 80%, while tox tree and test had accuracy about 66 to 77%. So I think if you are getting a accuracy of more than 75%, it's good. And additionally, QSR toolbox and tox tree had accuracy of 90% and 69% for CAR-C endpoints. I'm not going to show you the detailed results because this poster has still to be presented in September in the Eurotox conference, so I don't want to disclose all the results right now. We also have done a lot of work in the in silico domain in other and presented that in other conferences also, like we did present in the SOT 2023 meeting in March, where we presented genotoxicity and carcinogenicity prediction of drugs and chemicals using various tools like QSR toolbox, direct nexus, and test. We also, in that SOT 2022 meeting, we presented work on prediction of mutagenicity, genotox, and CAR-C of drugs and chemicals using direct nexus. This I have already, okay, sorry, this is copied into the places. I have shown one of the posters, so this is another thing which we have submitted to Eurotox. So we evaluated the phototox potential of various solvents used which are used in the in vitro tox assays, in vitro phototox assays, and we compared the toxicity by in silico prediction as well as in vitro 3T3 NRU method. We have also presented earlier in 2022 in silico analysis using direct and SARA for nitrosamines, and in silico toxicity prediction using direct nexus for skin sensitization, phototoxicity, and hepatotox and in vitro hug inhibition. So what I want to say is we are very passionate to do good science, to do good work and publish it or present it, so that others, especially regulatory agencies, can accept it. And if you are interested in collaborating, so if you have some ideas or where we can work together, or if you have, suppose we have these tools and you are having a different set of tools, so then maybe we can use a same data set and run those compounds in various tools and then come up with some good data. So that will be quite useful. So if you are really willing to do this, so we are always open to collaborate as a scientist. So this is all from my side. So how much time is left, Dr. Ankita, so I can take some extra slides, or is the time over?
Dr. Ahuja, unfortunately, the time is over. We're saving some time for questions from the audience. So,
okay, so then we keep it now open for discussion. I close it now.
Thank you so much, Dr. Ahuja, for your presentation and for the excellent demonstration of the in silico tools. I'm sure this is definitely going to encourage the industry, the people who have not yet exposed to in silico or still apprehensive about using these tools, to definitely give this a try. We have a lot of questions for our speakers, and we are going to try and take as much of them in the next 20 minutes or so. We also have Dr. S.K. Jain, who's the Senior Consultant Toxicology from the Central Insecticide Board and Registration Committee, join in as a panelist today. So this is an opportunity for all the industry people out there to quickly pour in as many questions as you want for Dr. S.K. Jain to clarify. Okay, so moving to the first question, this is for Dr. Bharath. Dr. Bharath, can you just give us a brief idea of how these in silico models can be integrated with other NAMs to eventually avoid animal testing? You're on mute.
Yeah, thank you very much for a very relevant question. And in the interest of time, I could not cover it. So I take it as an opportunity to explain the concept here. So when it comes to the lamps, we have multiple animal-based experiments, particularly developmental as well as repeated dose toxicities, where we have an involvement of lot many animals. So instead of going for a experimentation always, particularly in case of impurities or intermediates, it's better to always offer a in silico tools, so you will have a fair idea like what would be the possible result. And if it is really negative, not necessary to go for the prediction, and you can also use it in a screening platform.
Okay.
I just want to add here, Dr. Ankita, that there is already a OECD 497 guideline on the skin sensitization, which came around, I think, 2020. So it describes that how you can use in silico tools. So there they have validated two in silico tools, which includes direct and OECD QSR toolbox, and they have given an integrated testing strategy where you can use in silico tools along with the in chemico or in vitro tools like DPRA or HCLAT to determine sensitization, and this tool can replace in vivo testing. So for skin sensitization, this guideline is already there, where you can use QSR as well as in NAMs in combination. So this is already there and people can use it.
Yeah, actually here, when it comes to LLNA EC3, guinea pigs are used, and they are again a large animals. And whatever the auto workflow or defined approach mentioned in this 497 can significantly reduce the usage of large animals like a guinea pig.
Thank you, both of you. Next question is for Dr. Ahuja. So can you please expand a little bit more on how you can possibly combine multiple QSR tools for prediction of toxicity instead of just relying on one source?
Yeah, so there are already a lot of publications available in the public domain, and these came before this ICH M7 guideline, because then at that time there was a lot of validation work ongoing around 2010 or so. So they show that if you use QSR tools in combination, they are more predictive as compared to a standalone tool, and that is why the regulatory guidelines say that you have to use two QSR tools in combination, but one is an expert tool based and the other should be an statistical based, so that increases the accuracy of the prediction.
Yeah. To add to Dr. Ahuja's point, when we use more than one tools or more models, we will have a more diversity of molecules covered, and so that our range will be more, prediction range will be more, because both the data sets will be containing different molecules. So our prediction range will be wider. So we can cover the diverse molecules. So that is another point we can add here.
Yeah, I agree with you, Dr. Bharath. Thanks for adding that.
So Dr. Bharath, next question for you. What is the basis for selecting an analog, and is there any reference available on how much similarity the chemical should be when compared to a chemical of interest?
Yeah, there is no any such a hard thumb rule for the selection of analog. It is always depend on applicability domain, as Dr. Ahuja was demonstrating in a Vega report. So you could see the applicability domain where the structural similarity is one point, and at the same time you could see two items in that: what is the actual activity and what is the predicted activity within a data set, whether we are getting the same predicted activity or actual activity or not. If that is not accurate, by then having a good analog is of no use. So it all depends on applicability domain rather than just looking at the analog similarity.
Dr. Ahuja, do you want to add?
Yeah, I just want to add here that when we search for analog, definitely we look for some criteria. And if you have a sufficient number of analogs available which have data in the public domain, so then we look for like more of a structural similarity, which should be at least more than 75%. So then we can confidently say that this is more similar, and this structure similarity is based on a Tanimoto coefficient. So some of the softwares also give that.
Yeah, in Vega we have a similarity index. So that will give us we have accuracy index as well as similarity index that actually guide us properly.
Next question I see here is for Dr. Ahuja. So can in silico models be used for predicting toxicity of new molecules that may be data poor without having to do any animal testing? Is that possible?
Yeah, so we do it for new molecules, like in pharmaceutical domain, definitely use that. But as I said, all the endpoints are not validated. So like for mutagenicity, which is well validated, you can confidently use that, and we do that. So suppose you have 10 hit molecules, and you just want to sort out one or two from them. So we use in silico tool for predicting mutagenicity. And since other endpoints are not validated, and also other endpoints are quite complex also, like if you say systemic toxicity or hepatotoxicity, so there the confidence is not much. Although we have also worked on some of these endpoints, and there are publications available also, but then still they have to be validated by regulatory agencies, so that whether they will accept it or not. So we cannot right now say that we can replace animal testing at for those endpoints. But yeah, for the mutagenicity or genotoxicity, definitely you can screen them in the early phase, and on the basis of that you can define your testing strategy by selecting a limited number of compounds from a bigger domain.
And Dr. Varun, there's another question for you which says, if a compound has variation difference in result for a particular endpoint in two different QSR tools, how should the results be interpreted?
Yeah, so I presented this in the first slide. So you should know about your software, about its database, and also where it is applicable. So if I suppose if I'm studying a pharmaceutical, then I'm more confident using direct, because I know that the database is very good for pharmaceutical. But when I'm using OECD QSR toolbox, I'm more confident using analyzing a chemical there, because I know the database is good for chemical but not for pharmaceutical. So yeah, you should know what is the database of the software so that you can use the right compound for analysis. Anything you want to add here, Dr. Bharat?
Yeah, this is actually a tricky question, and this is a tricky situation. Two different tools giving two different results. And as you have rightly mentioned, it all depends on our data set and the coverage of a chemical class, particularly when it comes to pharmaceuticals, we have a different range of molecules, and compared to that, agrochemicals are totally different. So whether it has a coverage of that class of chemical is a very important information we will have to look for.
Thank you. The next question is actually for both of you. Dr. Bharat, you can go first. So is there any instance where predictive toxicity endpoint is negative, but in animal or the real experiment, it would be positive? If so, what should be the next step?
It is very much possible because it all depends, as I have mentioned, it all depends on our data, whatever we have. That's why in a limitation, very firstly, I have told like it is data driven, because some class of molecules might not be explored for that particular toxicological endpoint. So if it is a case, we would end up with a situation like this.
So,
Yeah.
Dr.
So
Ahuja,
yeah, so as none of the assays are 100% predictive, so QSAR is also like 72%, 80% predictive. And similarly, if you take any in vitro assay or in vivo assay, so people rely very much on the in vivo assays. But if you see in the literature, in vivo assays are also only predictive, like about 70% or so. So definitely it can happen that when you analyze something in QSAR and the results can be different in the assay, in vitro or in vivo assay. So first thing is, as a toxicologist, we don't know much about the chemistry of the compound. So the toxicologist and chemist have to work together. So chemists know a lot about the structures and what can happen with them, and toxicologists know the toxicity. So we have to work together and see, and then definitely we have to take a call. So that is why QSARs are just predictive, and other assays are like in vitro, in vivo assays are more reliable. But then it also depends on the endpoint which you are testing for. Like mutagenicity, you can be more confident as compared to a systemic toxicity where the assays in silico assays are not validated.
Okay. So, a general question, can you both give us a little bit of insight on your experience of making successful in silico submissions uh, globally? I mean, to what extent do you think these are readily acceptable by regulators globally? And how do you think, I mean, do you have any experience of making some submissions in India? And if you can just give a little bit of insight on that.
Yeah, we have a clients from different part of the world, and we have really given them a service for different regulatory submissions. And so far, whatever the predictions we have made are really appreciated by the clients, and it has served their purpose. And even regulators in India are currently now looking forward for the QSR predictions, and probably in future, even in India, we may receive the good appreciation for QSR studies.
So yeah, thank you, Dr. Bharat. So I can say that so for in the pharmaceutical domain, the predictions in silico predictions are very well accepted for pharmaceutical impurities. So all the global regulatory agencies, FDA, EMA, India, or other countries, they accept it. So we have submitted a lot of reports. I think I would say about 100 reports to various regulatory agencies from using QSAR. So they accept it very well. They are also accepted for pesticides in the European region. I can't say how much it is accepted in India because I don't have any experience with them. But yeah, in European region it is accepted, especially the QSAR reports for the genotoxicity prediction, especially for the metabolites that I also showed a case study on that. So they are accepted there. Yeah, so acceptance is definitely there. So I think in India it has to increase. So we have to work together, the regulators, scientists, and the industry to increase the acceptance.
Thank you. I think with Dr. Jain on board, I think the industry, I mean, all the audience would be really, curious to listen of what the situation is in India. Dr. Jain, can you just give us a little bit of insight into whether such approaches are have been considered for regulatory submissions in India? Dr. Jain, are you there? I see him online, but okay, I think we'll probably wait for him to answer that question. We can just take on some other questions. Okay, so there's this question which says, I have LCMS data of many metabolites of plants. Can we use it for prediction of toxicity? I mean, whether only LCMS data will support. I'm not sure if this is completely relevant for today's session.
Yeah, yeah, please
Sorry.
go ahead, Dr. Please.
You can start. I will add later.
Yeah, all it is required to do the QSR based prediction for any toxicological prediction is a structure. Until and unless you have a structure of a molecule, you can always go ahead for a toxicity prediction for your molecule. But having only the mass, it won't help us.
Yeah, I think you have answered, Dr. Bharat. I agree with you.
Yeah.
So LCMS identifies only the mass. So after that, the person has to look what is the compound, and once you know the compound with the structure, then you can do the in silico analysis. Dr. Ankita?
Sorry, I forgot to unmute myself.
I
The next
thought we
question.
are alone.
No. Dr. Varun, is there any difference in the weightage given to the results obtained from QSR tools, read across, or structured alerts if the results obtained from these tools are completely different?
Pardon me, can you please repeat your question?
Just a sec. So it says that when you have different alerts from these different tools like QSRs, read across, so how do you decide on which alert to give weightage to? I mean, how do you take the call?
So you are asking about two different approaches. So one is in silico tool is definitely used when you have a known compound. So suppose I used endosulfan. So when you will run it in the in silico tool, endosulfan is a known compound. So suppose if you need have an unknown compound but you have the structure, so you will run it in the in silico tool. Read across is done when suppose your unknown compound is not having any data, and then you find a analog or surrogate for that, which is having public available data in the public domain, and then you use it. So now if you are running these two in the in silico tools, so definitely if they are two different compounds, then the structure alerts will be different. So I can give you an example. So suppose we have an impurity, and we run that and we search for an analog which might be a known compound, which might be suppose a pharmaceutical for which the data is already available. And when we run the structures in the in silico tool, so as I said, for the analog, you should we always look for some similarity. So we try to see if the similarity is more than like 75% or so. Sometimes we see 90% similarity also. So the thing is, maybe when it is more similar, the structure alerts may be quite similar. There will be definitely a little bit of difference between the two, but then you have to justify that, okay, for the analog for which the data is already available in public domain, so what are the toxicities which are there as per the data available, and for the unknown compound the structural alerts are there, so how much they are matching. So it will not definitely be 100%, but it should be match, because you are doing a read across and there should be some similarity, so that is why it is an analog. You want to add something, Dr. Bharat?
Yeah, thanks, Dr. Ahuja, for giving a very basic for this particular question. And I would like to tell that this QSR as well as the read across are a sequential process, I can say. So initially we go for a QSR. If we get a hit or there are alerts over there, and if there is no data gap, then we will have to take the QSR predictions. Otherwise, if there is this, we usually call as a measure values, and if it is not available, if there is a data gap, that data gap can be filled using the approach called as read across. So where the source data will be referred for a desired information, and from that the data will be taken for the target molecule. So these are the sequential process.
Thanks.
Great. Thank you both. I think we've almost reached the end of our presentation. We have many more questions that we want to get to, but because of the time limitation, we can definitely get back to the people who reached out to us with questions, with answers. Dr. Ahuja and Dr. Bharath, just something on behalf of Dr. Jain. He was there for both the presentations, but unfortunately, he had to move out to attend another call. So he asked me to congratulate both of you. It was an excellent presentation, very crystal clear and crisp. And he did ask me to say that, looking at the presentation and the kind of evidence you guys have given, this is something definitely that CIBRC would be willing to consider if the industry is making in silico submissions. So thank you so much.
We will be really thankful if that happens. So thank you.
Yeah. So yeah. So we'll just proceed with conclusion. Thank you everybody again for joining today's webinar, and to our both the speakers here. If you have any questions, comments, please do feel free to write to me at my email ID. And as the webinar ends, there will be a pop-up to answer a few survey questions on today's session. So please do spare a few minutes to answer those. We'll really appreciate your feedback. And please enjoy the rest of your day. Thank you so much. Thank you, everybody.
Thank you.
Thank
Bye.
you. Bye-bye.
Thank you.